My topic today is on Paracentral Dogma, okay, supporting the central dogma with sugar cause. I do the next one. It doesn't work. Sorry to take your time. That one doesn't work now. Okay, good. Thank you. Yeah. So I will start with central dogma from DNA, protein, RNA, protein, and every people knows here, the transcription, translation, and now we understand, the protein and the lipids are glycosylated. And recently, another breakthrough is the RNA can be, glycosylated. So in such a scenario, and the people will ask, between the lipids and glycans, the sugars, who talks, who listens, and also their interactions with proteins. So all this, okay, and to address this research question, our team proposed, the Paracentral Dogma. Is there another law exists to contribute the central dogma for such, a regulation? And I'd like to start, with this study, a very interesting one from the, Caroline Bertozzi's group. Okay, they got the Nobel Prize in last year, and this paper tells a lot of information. First, I think, classically, as every people understand, the glycosylation start from ER, then, to the Golgi apparatus for the modification, then, start their functions, all this complicated process. I will show this, in my next slide. But this work first time told us the RNA can be glycosylated, and also the glycosylation happened in the cytosol. So this is very interesting, very challenging. And on top of that, the RNAs can, like, the lipids protein, glycosylated lipids protein can express on the cell membranes and then interact with the receptors. So this gives a lot of meanings. Here, I think, for example, the 293 tRNA glycans, and you can, if people working on virus from the previous talks, and this enzyme heavily involved in the RNA replication, okay, the virus, duplication, all these things. And then the second one, H9, HESRA, this one, if people working on the stem cells, also this is the enzyme, RNA involved in the temple spiral, all these cell, stem cell development. And the last one, HeLa RNA. Okay, these HeLa cell lines, every people knows. Okay, if you do the cellular biology, so this is the the cell lines used every day for us. Okay, never dies. Okay, so this raises another question: what are these? What's the difference? All these glycosylated RNA with the native form. Okay, so all these questions, and also gave us a lot of meaning. Okay, the potential application in science. Okay, in glycomedicine. And based on these principles, from these slides, you can see how the glycans, processed in the ER and Golgi apparatus. But make it very simple. Based on the previous talk, the genetic part. Okay, the genetic, the DNA can be as a static markers for disease diagnosis. But for these glycans, they are dynamic, okay? And my team has been contribute to this from this perspective. Can the glycans, we use them as the dynamic markers for the preventive, predictive, and personalized or precision medicine. So in my next talk, I'm going to give you several examples. And this is the example of IgG. And today is the keywords, okay, one of the keywords for our talk. And you can see from here, okay, actually glycosylated IgG through the type 1, type 2 receptors, then where the all the executing molecules, for example, interleukin 6, TNF alpha, and on the other pathway, the interleukin 33, interleukin 4, for all the the balance, the equilibrium between the inflammation and the resolution of inflammation. This, pro anti-inflammatory glycans actually give us a very powerful battery of biomarkers for disease prediction, for precision medicine. For example, here at the right corner, you can see, for example, 2 glycans. From the structure, you can see they are changing now. For example, the first one, the galactose, they removed. Then they further remove the sialic acid and become the simple one. Okay, then from the quantitative analysis perspective, the level of these molecules changed. You can see this curve now. So this is very interesting. This gave us a profiling, dynamic profiling to tell a disease, a status. For example, from this, the blue column here, this is the window opportunity, or we can name this status as suboptimal health or subclinical status. Then the curve goes up. Then a lot of such simple molecules, glycans in our bodies. Okay, then this actually tell clinicians, okay, this disease, for example, rheumatoid arthritis, okay, is in the active phase. Then we put the treatment, okay, for the monitoring, and it seems the drug works. Okay, then back to the normal. So in such case, really can tell us, the real life, real time series of the disease development from the early development, treatment, and to the prognosis. Okay, all this make us so interested in these glycan markers. And this is the work we did first time, and I named this as the glycan fingerprinting for Chinese Han populations first. But I think fingerprinting never changed during the lifetime. So I changed the term. This could be the profiling, or the glycan composition of the Chinese Han populations, and also collaborate with the Gordon's group. We validate this, the profilings of Chinese Han population in the Caucasian populations. And the more important things, we focus all these glycans as a biomarker to the, the clinical health measures. We are not talk about disease. Here we talk about, the health measures, how health measures, for example, the blood pressures, the BMI, also the fast glucose levels. All this could be, very interesting. Every day, when we do the clinical check, all these are the major biomarkers for us to tell us how healthy we are. So they linked with all these glycans. Okay, so this gave us a battery of markers for measuring health. And for example, here, and also, like Gordon mentioned, we developed, the biomarkers for the, the biological age. In another word, we want to tell, how your biological age different from your ID card age. And recently, another talent from my team developed another one. We name it as the cardiovascular age. So in that, you can see we want to tell you how young or how old is the heart. Okay, so this is another interesting perspective we developed. Okay, and this work published by engineering. Okay, recently. And then back to the keywords, the topic of my, the title I mentioned, Paracentral Dogma. So what exactly I mean? Okay, and this is from one of my postdoc. Okay, a very clever girl. Okay, say, Wei, do you want to tell the audience about this information, genetics, like you already talk about all this genetic part is the letters. For example, eat. Okay, and this. Then, okay, amino acid. Then they change the meanings. For they can be different meanings for these, three letters. For example, eat, ate, add. Okay, so all the meaning changed. And then what is glycan? They say, okay, this is the punctuations. Okay, so what exactly, in such case can, let's see these two sentences. Let's eat, grandpa. Then if we put a comma between, let's eat, comma, grandpa, totally different. Okay, so this gave a completely different meanings, right? So this question actually, alive and die. Okay, so Jesus, so this is exactly what I mean. And here, I would like to elaborate a little bit more. So in today's talk, and this keywords has been mentioned several times. For example, the glycotransferase. And in our eyes, these are the encoder. These are the writers. Okay, actually they just encode, transfer from the donor to the acceptor, and they write language. Okay, all these glycans, okay, play a fundamental role. Okay, for example, the skeleton, of the cells. Okay, all these proteins involved in this process. And also from here, you can see another keyword is the glycosidase. And these guys actually, like the scissors, okay, they erase, they cut off all these unnecessary ones, okay, they change the glycans, okay. So this and the modification of process of this glycosylation. Then naturally you got another interesting molecules. This mineral-binding lectin has been, mentioned several times in the previous talk. And these are exactly the readers. They recognize the virus, the parasite, the bacteria, the attack from, our outside of our cell, and also they read, the glycans actually attached to the proteins in our cell. Okay, and from the two perspective, you can see they themselves can be used as a drug actually to protect ourselves. And on the other hand, they can be used as a biomarker, okay, to tell, for example, the autoimmune disease, okay, rheumatoid arthritis and the SLE, for example. Then on top of this, how about the actors who do the job? Okay, so this is another thing here. Okay, for example, the IgG glycosylation. Now we fully understand this primary sequence, the secondary sequence, the tertiary sequence. All these, very different pathways. For example, antibody-dependent, cellular cytotoxicity, and also the CDC's complement-dependent cellular toxicity, and also the mineral-binding lectin pathways. All this involved. Okay, then you talk about the immune cells. Okay, all also involved in this process. So we have more and more pictures. The glycans not just as the classically as a group of molecules involved in metabolic. This metabolic things, okay, is one part of the the glycans. Actually, here they store the information. Okay, they interact with proteins, with RNAs, with the lipids. Okay, these are the glycan language. So I use the word glycomedicine in recently, this paper published in engineering. Actually, since the publication of this paper, many scientists from different countries actually write the editorial, ask, as a letter to the editor, debating on this, and I'm so pleased to see this. Okay, there are, since it's online last year, and there are over 26 public citations now. Okay, it's very amazing. Okay, I hopefully to see some more debating on this. And this just to do a promotion for this special issue. All the invited speakers, except Longgang, joined us later. Got the invitation. They contribute to this. Today's speakers contribute to this, special issue, and co-edited by Professor Yang. And I want to mention Professor Yang is the chief editor for the the medicine section of this engineering, and I'm the chief editor for the special section on the topic of glycom and glycomedicine. So we are trying to organize another one in year 2024. Okay, and this is the review. Okay, mini review. I first time proposed the Paracentral Dogma ideas. If you have time, interest, have a read of this paper. And also, interest after the publication, and I got the invitation from the Frontiers in Immunology. Okay, the chief editor asked me, Wei, can you elaborate, follow up the publication, the Paracentral Dogma ideas you published in engineering? Can you organize a special issue in Frontiers in Immunology? Okay, actually, this paper also just published. Okay, hopefully you have time, if you're interested in this topic, have a read. And also in my team, and to prove this, and in my mind, okay, there are postdoc PhDs, and my question to them, they propose, for example, pseudo time analysis versus real time, the single cell sequencing, and to try to answer the complicated questions to support the Paracentral Dogma. I'm looking forward, okay, to see the outcome. And also, I want to do a promotion for this, the Human Glycom project, also lead by the Professor Tan Xiri, okay, the dean of the Central University Medical College. And now we have Peking University Zhuanghui and Tsinghua University Chinese Academy of Science, and the Chinese Academy of Medical Science, Harbin Medical University, and Shandong Capital and Henan University joined us, and now Zhongshan and other universities, they are in the process of joining us, okay, for this Human Glycom project. And we are also very lucky to, we have all the, 4 overseas speakers, okay, they have agreed, to be the members of international consultant committee for our Human Glycom project. So under the Human Glycom project, so we will start this Human Glycom project China very soon. So this is, one example, the platform we provide, and for our collaborators, we are going to organize, a platform for the data analysis to support the scientists for our collaborations with the data mining, bioinformatics of the glycomics data analysis. And also, I like to mention this machine and collaborated with the Edelman, okay, from the Max Planck, okay, and also supported by the Shuoyuan Jin. Actually, we are developing the first glyco decoder. Actually, it's a sequencer for glycol sequencing. If you have time, whatever, you can come to Shantou University, Henan University. We have our first machine there, okay, to show you, okay, how this will apply to the human glycom China approach. And also, I like to thank all the peoples involved in this effort. Okay, this collaboration, international collaboration, make us all the planning the project possible. Okay, within a very short period of time. Okay, thank you so much.
工程创造人类美好未来。